Hi, my name's Scott Edwards, and in this second lecture we'll be continuing our tour of phylogeography, the exciting field of linking genetic variation with geography. And in particular, I'll be telling you about how I have applied phylogeographic principles to study the biogeography and evolution of Australian birds. Australia is an amazing continent full of amazing animals. And here we're going to use genetic variation to understand the history of those populations and species. So what we're going to first talk about is the context and the particular biogeographic setting of the problems I'm studying. We'll go to the savannas of northern Australia and look at a region called the Carpentarian barrier, which has been very important for producing biodiversity in Australia. We'll then talk a little bit about the kinds of molecular markers I'm using in my study. There are many ways to measure genetic variation, and we have chosen to use so-called sequence-based markers as opposed to markers called microsatellites, and we'll talk about some of the advantages and disadvantages of those two markers for looking at phylogeography. Finally, we'll move on to the fun part, which are the case studies. These are applying the principles to specific groups of Australian birds. Each group has a separate twist to it. The first three groups will be focused on this Carpentarian barrier, and the last group, which are the Zebra Finches, will be a separate but no less interesting biogeographic situation. And we're going to use genetic data to look at several different aspects of population history. We're going to look at population structure and gene flow, the movement of individuals between populations. We're going to look at divergence times and whether different groups of species have diverged at the same times or at different times. And then finally, we'll look at using many loci that are linked together on the same chromosome. And we'll learn how this sort of linkage can give us extra information about the history of populations. So, why have I decided to work in Australia? It's not just a fun place where you can meet Crocodile Dundee. Australia actually is extremely important and useful for understanding phylogeography. It turns out that the Australian environment has changed very gradually over the past 15 million years. Unlike the northern hemisphere continents like Europe or North America, Australia never had extensive glaciation. And so we never had a situation where the glaciers had dominated the continent and then receded, which of course has been the main driver of genetic patterns in animals living in Europe and North America. Australia had a relatively stable environment that changed very gradually. And as a result, you can see that the ranges of different groups of birds are remarkably similar, sort of a chock-a-block pattern where in two separate groups, their ranges will add up to those of a third. So here you can see, for example, on the top row, ranges of brown and black-tailed treecreepers, how if you put those together, you sort of add up to the range of a completely unrelated group, the babblers, the gray-crowned babbler. You also see the black-throated and long-tailed finches in the middle row. Those ranges add up to those of a completely unrelated species, the red-backed wren. So we'll be looking at all of these groups to show how their histories have been similar and in some cases different. Now, the Carpentarian barrier is a region of northern Australia that has had a lot of environmental change, and it's a region where related populations and species meet and in many cases have diverged into separate species. Why have they diverged here? Well, it's possible that there's been a lot of desertification, very aridification in this region, such that animals can't live there but are separated to the periphery. It could also be that there's been intrusions of the marine realm. The sea has actually made a finger into the interior of Australia and caused populations to split. Of course, the region in this part of the world has been very complicated. Australia, of course, was connected to New Guinea to the north on several different occasions, such that there was a single landmass. Now, New Guinea is, of course, an island separated from Australia by the Torres Strait. But nonetheless, this Carpentarian region has been a very important region for evolutionary change in the Australian continent. And that is borne out by these regions, these trees you see at the bottom of the diagram. These are called area cladograms. They are essentially summaries of the relationships of different populations around Australia. So you can see, for example, in the area cladogram on the left part of the screen, we've got several regions around the periphery of Australia. You've got KP, which stands for the Kimberley Plateau. You've got AL, which stands for Arnhem Land. You've got on the eastern part of the continent, you've got EF for Eastern Forests, AP for Atherton Plateau. You can see how all of these regions are related to each other in a genealogical or a phylogenetic context. And what these are essentially predictive tools. They are summaries of the histories of many different animals and plants, and we can use them as tools to predict what the relationships of populations will be based on species that we haven't yet studied. Most importantly, you can see how the barrier B, that Carpentarian barrier, is the deepest split on that area cladogram on the left side of the screen. What you can see is that the barrier B is an extremely important barrier, the most basal split in that area cladogram on the left. And this means that it's been a very important driver of diversification in Australian birds and other animals and plants. We've spent a lot of time running around Australia. It's a great place to work. The animals are fantastic. And this slide hopefully illustrates how phylogeography work takes a lot of time and a lot of travel. We've got to sample different populations. We've got to look at many different regions of individual species as well as many different species. And you can see how you can easily fill up several decades with this sort of travel. It's a lot of fun, and you learn a lot about the people and the places where you're visiting. Turns out the Australians themselves are also an interesting lot. As you can see from this sign here, shut the bloody gate. They're very direct. They don't like to beat around the bush. Much of our work is done on very large privately owned stations where they're running cattle or sheep. These stations are often biodiversity reservoirs. They're very important for harboring populations of animals and plants. And so we always make sure we shut the gate so the livestock don't run away. Now, I want to give you an example of an early phylogeographic study I did on a bird called the gray-crowned babbler. It's very widespread in Australia. What you can see here is that we've sampled several populations all over the eastern part of the continent as well as the west, and you can see that Carpentarian barrier there. These birds, it turns out, are divided into subspecies. They're not completely different species, but they're definitely differentiated across that barrier. So we sampled widely, and just to sort of fast forward, I can show you some of the results of a survey of a molecule called mitochondrial DNA. We mentioned this in lecture 1, where we made a comprehensive gene tree for all these different populations. And as we course through this gene tree, you can see how the populations are related to different geographic areas of Australia. This is really the essence of phylogeography. You can see there's a group of lineages up in the north in Cape York and southern New Guinea. You can see a group in the central part of the east coast. And then you can see a cluster in the southeast. And this is really very marked geographic structuring in this species. Similarly, in the west, you can see a broad group over the top end of the continent in the north, as well as a clade in the northwest. This is great evidence for strong phylogeographic structure. Now, mitochondrial DNA was the workhorse of phylogeography for a long time. It's a very important molecule for phylogeography because it changes very quickly. It's got a high mutation rate. We can get a lot of variation within a species. On the downside, it's only one molecule. It really only tells one story of what is really a collection of stories, each told by a separate gene. All of the genes in the genome, it turns out, have correlated histories. They're similar to each other, although not identical because of the vagaries of population genetics and stochastic forces. If we only looked at mitochondrial DNA, we would really only be able to say anything about that particular gene. What we'd like to do is to look at genes in the nucleus as well and get a broader view of what the actual populations of these birds are doing. So that's the reason why we've moved on to looking at additional genes in the nucleus. Now, some of the models that we'll be using are specifically designed to take in information from many different genes, not just one gene. And what we'll do now is briefly review some of the different models that are typically used in phylogeographic research. So here's a basic model. It's a model of population structure. So we're going to think about different populations connected to each other partially by gene flow, by movement of individuals between the populations. Under this model, we can also estimate the sizes of the populations and the amount of genetic diversity within each population. So this is a very important model if you're interested in how connected populations are to each other. And it's the software that we used called MIGRATE is very versatile, and it can estimate each parameter independently provided that you have sufficient data, sufficient number of different loci. We can estimate the size of the populations as well as the amount of gene flow between them. Another population model which is commonly used is so-called isolation migration model. In this model, we can imagine a single ancestral population, which you see at the bottom of the slide, splitting into two different populations. Each of these three populations, the one ancestral and the two descendant, have a population size, an effective population size, as we learned about in the first lecture. There may be gene flow between these two descendant populations, and in fact, in recent versions of this model, there are many more than two populations. You can have any number. You can also imagine that we can estimate the time that this ancestral population split into two populations. Like the migration model we saw earlier, this isolation migration model uses information from many unlinked loci that might be on different chromosomes and have slightly different histories that are all correlated with each other. And you can see the parameters that we're using on the top. Many of these parameters we discussed in the first lecture. There's theta, there's divergence times with mutation rates, there's gene flow, etc. Finally, we can talk about a model that doesn't involve any gene flow. In fact, it only involves a series of isolation events, a series of divergence events. You might call this the pure isolation model. In this model, it's essentially the history of a phylogeny, a history of populations that have been diverging from a common ancestor. And you can see that these populations are splitting at various times. Those are the taus on the left side of the slide, 2 and 3. You can also see the sizes of the ancestral populations. You can see the theta, 1, 2, and 3. All of these are describing the history of the splitting of these lineages. And this sort of model, I think, is very similar in spirit to the previous two models we saw. Again, we're going to use multiple loci that are not linked to each other, that can be treated as quasi-independent, somewhat correlated. Now, we're going to be using a series of loci, a series of genetic markers that you might call sequence-based markers, or in our case, anonymous loci, These are essentially random bits of the genome that may not even encode a protein. They may just be junk DNA in some cases. Why do we like this? Because it turns out that junk tends to change readily. It doesn't get stuck in one optimum because it actually has something to do, because it actually has a function. We can focus on the variation in this junk DNA because it's relatively abundant. It's under no constraints, and most any mutation that happens will be just as good as what was there before. So this is very, very handy. Now, we like these sequence-based markers because we can measure genetic parameters very easily with them. We can, for example, compare nuclear DNA directly to mitochondrial DNA. They're both sampled by using DNA sequences. They're essentially the same units, the same type of information. We can talk about the extent to which these genes mutate one way and then back again to the ancestral state. That's a bad thing because it erases the history before it. We call that homoplasy. In nuclear DNA, when you look at it in terms of the sequences, that sort of backtracking and homoplasy is minimized. We can build gene trees, as we talked about in lecture 1. Gene trees are a very intuitive way to look at population history. And we can compare the diversities easily across different species. It's very easy to do. These are all in contrast to a very popular marker called microsatellites. Microsatellites are essentially repeated regions of the genome that we assay not by sequencing, but simply by scoring the number of repeats within a marker. Turns out that microsatellites can undergo a lot of back mutation. Homoplasy is very common. It also turns out that we look at mitochondrial DNA and microsatellites in the nucleus in completely different ways. It's like apples and oranges. It's hard to compare them to each other. Finally, if we focus on microsats, we tend to only look at the hypervariable parts of the genome. We get really an atypical view of what's happening in the genome as a whole. Whereas with sequence-based markers, we can take more of an average, which of course is what we want if we want to make statements about the genome as a whole and about the populations as a whole. So on the bottom, you see a challenge for phylogeography. If we define the loci that we're looking at by looking at a small subgroup, what we call a panel, and then assay those markers in a larger group, which is our actual study population, which you see on the right. It's not a big deal because new sequencing techniques are allowing us to look at the genome in a very unbiased fashion. So this so-called ascertainment bias, this bias that we incur by looking at just a subset of previously known markers, is pretty much going to disappear. So it's a very exciting time. Okay, let's move on to the animals. We're going to look at 4 case studies, each of which has a slightly different story to tell. We're going to first look at a group of birds called grassfinches, which are distributed up in the north of Australia. Then we'll look at a group of birds called wrens. They're not like the wrens in North America. These Australian wrens are completely unrelated. It turns out they're called wrens because they cock their tails just like the wrens here in North America do. We're then going to look at a group of birds called treecreepers, which are intriguing because they also have undergone convergence with species in the northern hemisphere. Treecreepers are unrelated to the creepers that we have here in the US or maybe in Europe or Asia, but they nonetheless climb trees almost like a woodpecker, and they're very, very similar in many ways to the creepers here in the northern hemisphere. Finally, we'll move on to zebra finches, which have a slightly different history. They're not separated by the Carpentarian barrier, but can tell us a lot about the history of the arid interior of Australia. And each of these studies was conducted by different, very talented students and postdocs in my lab. So here are the grassfinches. You can see there are 3 species. Poephila is the genus name. We've got the long-tailed finch, you can see there in the northwest, as well as the black-throated finch in the east. That long-tailed lineage is split into 2 separate lineages, so we have 3 in total. Now, what we did with this study was to collect about 30 different markers from the nuclear genome. And we wanted to look at the pattern of gene trees. Now, you might remember from the first lecture, we talked about a phenomenon called incomplete lineage sorting. That's a situation where the alleles are not separating out by species fast enough before the next speciation event arises. And the result is gene trees that look mixed up relative to the actual species history. We predict in this system that the eastern species, the black-throated finch, Poephila cincta, will be the first to branch off, with the 2 western ones secondary. And let's see what the gene trees say about that. So here you can see 30 different gene trees, and on their tips they've got 3 letters, A, H, and C. The A stands for Poephila acuticata, which is in the northwest. H is for hecki, also in the northwest. And the C is for cincta in the east, the black-throated finch. Now, you can see on the top row that those gene trees, that are about 15 or 16, those match that prediction that I told you about in the previous slide. In those gene trees, the eastern lineage, the black-throated finch, cincta, is the first to branch off, with the 2 northwest ones branching off secondarily. All's well and good so far. But then you look at the next set of trees in the middle and the lower row, and it turns out those have different patterns. In the middle row, you can see that the cincta allele is actually more closely related to one of the western clades, the acuticata one, the long-tailed finch. In the bottom, you can see how it's the hecki allele as closely related to the cincta allele. Very potentially confusing. Doesn't seem to match the population history. Of course, as we talked about in the first lecture, we can reconcile this by thinking about the time it takes for alleles to sort out by species if speciation has been very rapid. And it turns out we can take all of the information from all of these gene trees and come up with a summary statement that is consistent with all of these stories. And as you might expect, this scenario will have as a characteristic very rapid and recent speciation events, because that's what causes these conflicting gene tree patterns. So here is an analysis of that data where we've applied the pure isolation model. Okay? What you can see is a single tree of lineages at the top with the 3 taxa in there. And you can see we have a number of parameters on this tree. On the left, you can see the time that we've estimated for the divergences. MYA stands for millions of years ago. You can see the effective population size of each lineage in the past. Quite large populations, 350,000 for that basal ancestor, and 200,000 for the more recent ancestor. Of course, large population size is a situation where you might expect these alleles to segregate slowly. They won't separate rapidly because a large effective population size means that genetic drift is fairly slow. It's not changing quickly. You can also see the branching order of these populations. Again, not necessarily in conflict with some of those gene trees that had, for example, the H lineage and the C lineages next to each other. This is entirely expected if speciation has been rapid. And then finally, we can actually place confidence limits not just on the gene trees, but on the entire population history. So you can see we have a confidence in the species tree of about 0.88, which is pretty high. We'd like to see 0.95 or maybe even 1 for absolute certainty, but 0.88 is a pretty good, we're pretty certain of this result, and that's exciting. Now, a couple characteristics of this study. It turns out that the more genes we look at, the more accurate our estimates are. Each of these 4 panels shows one of the parameters in that previous slide. And so, for example, panels A and B indicate those divergence times that we saw on the left of the previous slide. Panels C and D indicate the population sizes, the effective population sizes of those 2 ancestors, the ancestor of the whole clade and the ancestor of the 2 descendant lineages. You can see in all 4 of these panels, actually really just 3 of them, A, B, and D, the uncertainty, which is indicated by those dotted lines, is decreasing as we add more loci. Okay? As we're adding more genes, from 2 genes all the way up to 30, our estimates are becoming more certain, and that's good. We want to get more certain estimates. Panel C, now this is the population size of that most recent divergence. I'll just go back for a second. You can see the theta AB there, right in the middle of that tree. That's the parameter we're trying to estimate, the population size of that ancestor. Turns out that particular parameter is a stubborn one. It doesn't seem to get more certain as we add more genes. And this is just a fact of many population genetic studies. Some parameters are difficult to estimate. Okay, so more genes means more accurate results. We also can see an example of what we talked about in the first lecture, wherein the gene coalescence overshoots the species divergence. Again, why is this? Because the ancestral species had a measure of genetic variation in it already, and it had a certain depth to its gene tree, even before there were any splits involved. What you can see is that the black bars show the estimates of the coalescence time or gene divergence in those 2 splits, the most basal split and the split of the 2 northwest lineages. You can see that those gene splits are quite a bit deeper than the actual splits that we're interested in, namely the population splits. You can see that, for example, on the left, the gene split is as deep as the Plio-Pleistocene boundary, which is about 1.8 million years ago. Of course, in reality, the split that we're really interested in, the population split, that we estimate to have occurred much more recently, on the order of more like 500,000 years ago. And so you can see why it's very important to distinguish between the gene trees, sort of the raw data that we collect from sequences, and the actual inferences of the population histories, which is a different estimate. All right, let's move now to the red-backed wrens. Very charismatic species. The males are jet black with a shock of red plumage on the back, and the females are much more drab. Now, the taxonomists had told us that in this particular species, it turns out that populations up in Cape York, in that finger sticking up in the northeast of the continent, those should actually be related to populations in the north and the west. Now, that's a conflict with that area cladogram that you saw earlier. The area cladogram, which you can see here on the inset, suggests, in fact, that those Cape York populations should be more closely related to the Atherton Plateau and Eastern Forest populations. So CY is our Cape York. You can see how it's related to New Guinea as well as all the populations on the east, AP and EF. So we've got a conflict. Area cladograms say Cape York's related to one thing. Taxonomy and plumage characteristics suggest that Cape York should be related to the northern populations. What is the answer? So again, we collected a bunch of different genes, data from different gene sequences. And what you can see on this slide is simply a measure of genetic diversity at each of those loci. We collected up almost 40 different loci, and what you can see on the y-axis is the different categories of loci. We have anonymous loci, we have introns, which are those spaces between the coding regions of genes. Little molecular biology for you. And on the x-axis, we have that measure of genetic diversity theta, or 4n mu. You can see how it varies quite a lot between different genes. Like we learned in the first lecture, theta is a locus-specific measure. Depends on the mutation rate. What you can see, though, is that we have a diversity of different markers to estimate the population history of this species. So what we did was we used a bit of software called Structure. And what Structure is able to do is to cluster individuals into larger groups. And you run the model depending on different numbers of groups. What you can see in the table above is that this variable k, that's our number of populations in each of these runs. You can see k might be a single population, might be 2 populations, it might be 3 or 4 or 5. It turns out in this particular study, 3 populations were the best explanation for this data. Now, and you can see those 3 populations in the 3 colors in the dots, which are sort of extracted from the map of Australia with the northeast, northwest, and central east populations. Now, what you can see in the diagram on the bottom is that Structure allows us to put every individual into a higher organization. You can see each column is a single individual, and that individual has some of its genome from one population and some from another. And those colors match the populations you see on the top of the slide. So what this tells us is that, in fact, one major population is consistent primarily of the populations from the north and west, okay? That's the yellow dots. The other population is mostly individuals in Cape York, the orange dots, as well as individuals in the central east, which are the purple dots. And this is really exciting because this suggests that it's rejecting the taxonomy conclusion. It turns out that Cape York population in the orange appears actually more closely related to the eastern populations than to the ones in the north and west. So we've been able to test that hypothesis here. We accept the area cladogram prediction and reject the prediction from taxonomy. So this is a classic use of phylogeographic data. Let's talk about another great group of birds, the treecreepers. These are the ones that climb trees like woodpeckers, even though there are actually no woodpeckers in Australia. And it turns out these are also, like the wrens, they're also what we call cooperative breeders. They have an interesting social system. They have helpers at the nest. Birds that could breed on their own, turns out they wait and help their parents raise the next brood. Why they do this is a bit of a mystery, but and you could imagine how this might influence their genetic diversity depending on how they breed together. Anyway, like the babblers, we've sampled them all across the continent. We then went out and looked at genetic data from a bunch of different anonymous loci. Here's some examples of the anonymous loci, the gene trees, which you can see are, each one is AL for anonymous locus. You can see that there are red lineages, which are the brown treecreeper from the east. You can see there are green lineages. These are the black-tailed treecreepers from the west. You can see that each of the gene trees is different. None of them are identical. They're all showing slightly different patterns. And nonetheless, one postulate of phylogeography is that if all of these loci are neutral, and they probably are neutral because none of these loci actually encode a protein, if they're all neutral, they should all record the same history. So even though the gene trees are different, we can imagine we're trying to construct an overarching story that accommodates all of the variation here. Okay, we can talk about a model of gene flow and isolation, the migration model that we mentioned earlier. We talked in the first lecture about a sort of a parsimony approach where we counted these interspecific coalescent events down the tree. Those are those block dots on those gene trees that we saw earlier. This approach was useful when it was first brought in the late 1980s. turns out it makes a number of assumptions that we'd really rather do without. For example, it assumes that our gene tree is known with complete certainty, which is rarely the case. It turns out that it's somewhat cumbersome to test different hypotheses under this framework. And the many different sources of uncertainty in population genetics are not adequately accounted for. So instead of that parsimony approach, we're going to use an approach called maximum likelihood. This is a more robust statistical approach that's embodied in software such as MIGRATE. And in this approach, the gene trees are estimates. We're not putting all our money on a specific gene tree. There's going to be some uncertainty around each gene tree. We can also estimate the sizes of each population individually, as well as the amount of gene flow between those populations. In the earlier models, we could really only assume that all the populations were essentially the same size. Finally, the maximum likelihood method is much easier in terms of hypothesis testing. We can rank different hypotheses and ask ourselves which one's the best explanation for the data. So we're going to apply MIGRATE to our Treecreeper data. You can see the ranges here of the brown Treecreeper in the east and the black-tailed Treecreeper in the west. You can see we've sampled all over the continent. Now let's first look at the population sizes. Here are our estimates of theta, or genetic diversity, within each of these populations. And what you can see is that some populations, such as the one in Weipa in the north, have very little genetic diversity. This suggests that their effective population size is relatively small. Probably not because mu is small, because we imagine that all these populations probably have the same mutation rate for these genes. What's probably small is n. Remember our equation 4n mu. Probably these birds are isolated. They may not be getting any new diversity from other populations. And as a result, their effective population size is fairly small. What you can also see on the west is a great diversity of population sizes. The Fitzroy crossing population size is quite big, perhaps because it's getting genetic input both from the north, from the Douglas Hot Springs, as well as from the south, the southwest, from the Fitzroy crossing region. I'm sorry, from the Newman region here. Regardless, the beauty of this model is that we can treat each population independently. Each one has a separate amount of genetic diversity. Let's look at gene flow now. Those are the arrows connecting these populations. You can see that in the east, in the brown Treecreeper lineage, we've got a fairly constant flow of gene flow across all of these populations, okay? You can see they're well connected by gene flow. By contrast, Weipa in the north is somewhat isolated. That's what the thin arrow means. It's not experiencing as many migrants as are the other populations. This might be leading to its low genetic diversity. In the north and the west, you can see that this northern swath really, all of these populations are fairly well connected to each other, okay? They're not significantly isolated from each other. By contrast, the Newman population in the extreme west is very isolated. It's got relatively low gene flow with other populations. So you can see how we can get a very nuanced picture of the genetic variation using multiple genes and a sophisticated maximum likelihood model. So now what we're going to do is talk about the population divergence times on the east and west side of the barrier. Now, the intriguing thing about these results is that the divergence time estimated for example, the Treecreepers here, is actually less than, it's about 300,000 years. It's actually less than the divergence time estimated for a species which is considered a single species, namely the red-backed wrens, okay? We would expect the Treecreepers perhaps to have an older divergence time. After all, they look quite different, as you saw in those slides. By contrast, the red-backed wrens are essentially a single species, albeit one that has diverged slightly. So this is one of the surprising results of this particular study. But what we'd like to do is to throw all of this information together and to ask the question, do we have a single divergence event going on among these three lineages? Or do we actually have three separate divergence events? This is the kind of hypothesis you can test with phylogeographic data. So let's look at that question. What you can see here is an analysis done by my colleague Mike Hickerson, where we've tested three hypotheses against each other. Those hypotheses are illustrated on the bottom of the diagram, and they are the hypothesis that there was a single divergence event, that's one, you see on the left. There are two divergence events, or that there were three separate divergence events for these three groups. Now, we're using a technique known as Bayesian statistics. We're not going to go into details here. Suffice it to say that going into the study, we didn't have any particular preference for one of these three hypotheses. And for that reason, we're going to say our prior beliefs equally rest on each of the three hypotheses. That's what you see in the open bars here. You can see that the probability is essentially equal for each of those three hypotheses. Now, in Bayesian statistics, the answer, or our estimate, is called the posterior. And that you can see in the dark bars. And the data suggests, in fact, that the hypothesis with the most weight is the one that suggests that there was a single divergence event across all of these lineages. This is intriguing because, again, some of the species, like the treecreepers, were very different, two different species, whereas the red-backed wrens were a single species. We might have expected different divergence times. Given the data we've collected, our best hypothesis is that there was a single event, perhaps intrusion of the sea or a desertification event that split all three populations at the same time. This is a really exciting result because it gives us a broader view of some of the forces that may be affecting Australian birds and other animals in Australia. Okay, we're going to move now to our last example of using multiple genes to look at phylogeographic history. And in this example, we're going to move away from the Carpentarian barrier to the arid interior of Australia. This is the true Australian outback. You better bring extra tires with you because if you get a flat tire out there, it may be hundreds of kilometers before you reach the next gas station. So we're going to study a species called the Zebra Finch. Very widespread and abundant species. Also a very important species for two reasons. One, it turns out that zebra finches are a very important model for studying what we call vocal learning, the ability of animals to learn how to speak. For that reason, zebra finches are actually good models for human speech. We can do experiments on zebra finches, of course, that we can't do on humans. And yet zebra finches learn their vocalizations just like human infants learn how to speak. So we can learn a lot about how humans speak by understanding the neurobiology and the genetics of species like the zebra finch. It's also important because the genome of the zebra finch, the entire collection of DNA letters in its chromosomes, has been decoded. There are about 1.2 billion letters in this bird's genome, and we know nearly every one of them. And this gives us a really unprecedented window into using the genetic variation to study phylogeography. So what we're going to do here is we're going to look not just at random genes, but genes that we know are close together or far apart on the chromosome of the species. This is the type of information that we can't get from most species on the planet because we don't know where their genes are located on the chromosome. Thanks to the Genome Project, we know, for example, that locus 1 and 2 are 2,000 base pairs apart. KB stands for kilobases, or 1,000 base pairs. And we know that locus 1 and 3 are, in this case, 10,000 base pairs apart. This information we didn't have for the 3 case studies we just saw. But as you'll see, it provides us a lot with very useful information. You can see in this study, we have actually gone fairly light on the numbers of individuals we've sampled. Just simply 8 times 4 is 32 in the mainland Australia, plus 12 in the island system of Timor in Indonesia. Turns out for many kinds of phylogeographic questions, again, it's not so much the number of individuals, the number of loci that gives you more information. So let's look at some of the patterns that we've seen in these, both the continental populations of zebra finches as well as the island populations in Indonesia. So let's look at genetic diversity. It's a standard measure of phylogeography. You can see nucleotide diversity on the y-axis, and you can see here for each of about 26 loci on the x-axis how much genetic diversity we have. You can see that in the Australian populations, which are in the black, quite a lot of genetic diversity. Those bars are really, really high. By contrast, the island populations in orange have much lower genetic diversity. In fact, for several of those autosomal loci and those loci called Z loci, these are loci on the so-called Z chromosome. It's like the mammalian X and Y chromosome. Birds have a Z and a W chromosome. You can see that for many of those loci, there's actually no genetic variation. None, zilch, zero, nada. Those are loci that show no sequence variation, even across all of those individuals. And this is important. This gives us information about the history of those island populations, as we'll see. The Z chromosome is interesting because it is present in 2 copies in males for birds and only 1 copy in females. And so this is like what we see in mammals, where the Y chromosome is present in just 1 copy in males and not present at all in females. Turns out that the effective population size of the Z chromosome, we expect that to be smaller than the effective size of genes on the autosomes. Why? Because there's fewer copies out there in the population. For every 4 copies of autosomes, there are 3 copies of the Z chromosome. So you can see that would reduce the amount of diversity. And that's partly why the diversity in the Z chromosome is lower than what we typically see in the autosomes. Okay, you can see here some measures of differentiation between the mainland and the island populations. You might remember our measure of genetic differentiation called FST. That's a fixation index. When FST is high, close to 1, it's lots of differences between populations. When it's low, close to 0, it's very little difference. You can see that all of these genes show slightly lower, sort of moderate levels of FST. You can also see that none of the genes show what we called reciprocal monophily, namely that the island and the mainland populations were clustering in different parts of the gene tree. In all of these gene trees, they're all mixed up. And again, this is a standard part of population history. Although it may seem complicated, it could be the result of a simple divergence event in the recent past. Let's use our model of structure to ask how we can place different individuals into population clusters. What you can see is a large number of individuals on the mainland which essentially are not genetically differentiated. They fall into a single large group that we can call the mainland or continental group. That group shows a remarkable amount of difference from the island group, the Timor samples here, which are like black and white. They're completely different genetically, even though they didn't show that reciprocal monophily. It was sort of a modest amount of differentiation. This structure method is able to pull out the differences between populations and really allow us to see the forest for the trees. So quite a lot of distinction between the mainland and the island populations. We can estimate from these data some information on the population history of both the mainland and the island populations. So on the top you see an estimate. This is again a Bayesian analysis. We get a distribution estimate of the effective population size of the mainland population. Turns out it's a whopping 1,300,000. That's a big population size. What that means is there's a lot of genetic diversity. As a point of comparison, let's ask what the effective population size of the entire human species is. All 7 billion people on the planet. Turns out we look as if, genetically, as if we're only 10,000 individuals. We're that genetically similar. The effective population size of humans is much, much smaller than the effective size of zebra finches. Who knows what the actual population of zebra finches is? It's probably, could well be 7 billion. They're small birds. There's a ton of them all over. The important point is that zebra finches harbor much more genetic diversity than do humans, and that's why their effective population size is so big. By contrast, the island population, which you see on the bottom, very little genetic variation. Our estimate of the effective population size is therefore much, much lower than in the mainland continent. We can come up with an overarching scenario of what has happened to this zebra finch using this isolation migration model that we talked about earlier. You can see how this population model is much simpler and straightforward than all those somewhat confusing gene trees. And so I like to summarize my results in terms of these population histories. What you can see is at the current day, at the top, you can see our effective population size of the current species, 1.3 million in the continental populations, 18,000 for the island populations. Using the isolation migration model, we estimated a divergence of these two populations about 2 million years ago, quite an old divergence. Perhaps more remarkably, we estimate a tiny, tiny effective population size for that island population when it first started 2 million years ago. Who knows, maybe a small band of zebra finches flew to the Lesser Sunda Islands, made it far enough to colonize and reproduce, but only a small flock. Of course, it may not have been 5,000 individuals. Remember, these are effective sizes. It may have been a smaller number. But it gives us a window into the history of this species. It tells us that there was a small group of individuals that colonized these islands a long time ago, and that since then these two populations have been experiencing independent evolution. So much so that they're actually, you can see the difference in their plumage. They actually look and behave differently. So this is a view of this species that we're able to get by looking at these many different loci. Okay, in this last bit, we're going to use the physical distance between genes to actually provide information about the population history. So we're going to talk about a phenomenon called linkage disequilibrium, and that's essentially the correlation of patterns between different sites in the genome. Okay? So we can talk about disequilibrium between sites that are one base pair apart, very close together, or we can talk about disequilibrium between sites that are very far apart, maybe even on different chromosomes. And in this case, we're going to color those cells that are showing a lot of similarity in their genetic patterns. We're going to color those cells black. Those are, if we travel down this triangle, those are going to be the cells that link two sites at different parts along the chromosome. By contrast, cells that are white or gray are showing very uncorrelated patterns, very different patterns. Now let's look at the pattern of linkage disequilibrium in the island and the mainland populations of zebra finch. You can see that in the mainland birds, we see relatively modest linkage disequilibrium. There's not a whole lot of black cells in that triangle. That means there's not a lot of correlation between these sites. By contrast, if we look at the island population, which you see here on the right, lots and lots of linkage disequilibrium. What is going on? Well, linkage disequilibrium can arise from many different processes, one of which is a small population, or a founder effect, when populations are established from a small number of individuals. That can cause a lot of the genetic similarity to be similar and cause different individuals to look the same, causing LD, as it's called, to increase. You can see that we see that in the island populations, perhaps because of this founder effect that we saw in the previous slide. By contrast, we don't see any appreciable linkage disequilibrium in the mainland population because that population is so large and it's very easy for the different sites to have different independent histories. So, could this be a case of what we call founder effect speciation, where a small group of individuals colonized the islands and then evolved into a separate subspecies or species? Well, possibly, because we know that founder effects can cause allele frequencies to differ. As we saw, the mainland and island populations were quite different. It can cause linkage disequilibrium to increase, as we saw, and it can create new gene combinations and potentially new material for evolution on which to act. So it may be that the founder effect in this species has helped to move the species along to different populations in the island and the mainland populations. And here are some examples on the bottom. You can actually see the plumage of these two populations are quite different. So we've been able to look at many different aspects of phylogeography, all of which are using different multiple genes, and we've seen how using more genes reduces our uncertainty of our estimates. We've seen how we might have some discordance in our gene trees, but nonetheless the population histories can be fairly straightforward. And we've seen how Australian birds are characterized by lots and lots of incomplete lineage sorting, but we've still got evidence for similar divergence times and similar population structures. So it's, despite the confusion that sometimes looking at individual genes can bring us, summarizing the patterns in terms of the population histories can be a very informative approach. And I would just like to thank the many people who have helped me work on these projects. Most of these people have been to Australia, and so they haven't been working in the lab all the time. They've actually been there to see Crocodile Dundee. So thank you very much, and I hope you've enjoyed our tour through Australian bird phylogeography. Thank you.